Well, good afternoon. Welcome back. Are you happy to be back? Did you miss your viruses? The answer has to be no, because you took them with you wherever you went, right? All right. We are in the home stretch here in this course. We got, what do we have? 12 lectures left. We're in the second half of the course. And you have one more exam and a final. So keep with it. I know it's a lot of stuff. But you'll be better off in the long run by knowing all about the viruses that are in you. I read a paper the other day where they sampled the fecal virome of one person for 2.5 years on a monthly basis. Daily would be a lot of shit, right? 10 to the 9th bacteriophages per gram of feces is what you excrete, okay? 10 to the 9th per gram. These are just bacteriophages. They actually didn't find any eukaryotic viruses in this person, which is really surprising to me. There should be lots of herpes viruses, adenoviruses, enteroviruses, so I think it was a methodological thing. But just think about that. And in the course of this 2.5-year sampling of one person, enough change, they could see a particular phage that they isolated early on, change enough in the course of time that it could be a different species. It changed enough, 3 or 4% of the sequence to be a new species. So the amount of diversity. Now the thing is, this is interesting because all of you have different viromes in your gut. And that's why, because they all change differently and randomly inside of you. Really interesting stuff. But today we're not going to talk about viromes. We're going to talk about more immunology. We have our last focused discussion of immunity. Today we're going to talk about adaptive immunity. And this is, of course, in our scheme of host defenses. We've talked about intrinsic, which is always present in the uninfected cell. We've talked about innate immunity, which is induced very rapidly by infection. Today we're going to talk about adaptive immunity, which of course is tailored to the pathogen, and it has memory, which isn't something that we talked about before. Now, just as an aside, we give terms, we try and classify things so that it's easier for us to study them, easier for me to explain it to you. So I give you intrinsic, innate, and adaptive, but there's always going to be an exception. Someone pointed this out last week. We talked about an intrinsic defense. CRISPRs are ancient intrinsic defenses, right? Everyone remember what CRISPRs are? There is memory in CRISPRs, though, right? The whole point of it is that the bacteria take the DNA that's coming into them, chop it up, and they integrate some of it into their host cell so that the next time they get infected, they can make an RNA directed against the incoming DNA and degrade it. So there is memory there, but we don't call it an adaptive response. So that's just kind of an example of how we like to put nice labels on things, but it doesn't always work. And often in class here, someone will ask a question which reveals why it doesn't work. So I figured I'd do the CRISPR before anyone preempted me. All right, today we're going to talk about the last stage of the immune response, which we began last time as a virus binds to cells. Here's an epithelial cell sheet. The virus enters and begins to replicate. Those cells produce cytokines. They sense the virus is foreign. They produce cytokines. Those attract a variety of immune cells, including dendritic cells, which begin to pick up the cytokines. They get activated. They also pick up viral antigens from the dead and dying cells. The dendritic cells mature. Most infections stop right there. The innate response can handle it. But if the virus continues to replicate above some threshold, which is probably measured in terms of the cytokine levels and the cytokines that are produced, dendritic cells go into the lymph node where they then instruct the adaptive immune system. And that's what we're going to talk about today. And adaptive immunity comprises cells and antibodies. The antibodies are proteins, as we'll see, that take care of viruses in many ways. And the cells include T cells that attempt to clear the infection. Before we talk about the mechanisms, I want to make sure we're all understanding what the cell types are involved here. Leukocytes. People get confused about leukocytes and lymphocytes. Leukocytes is simply a general term for white blood cells. All these cells on this slide are leukocytes. Monocytes, eosinophils, basophils, lymphocytes, and neutrophils. And then lymphocytes are a subset of leukocytes, specifically T cells, B cells, and NK cells. And one way you could remember this is that these have variable antigen detecting cell surface receptors. We talked a little bit about that last time for NK cells. We'll talk about it just a bit more today for T and B cells. So for the rest of the course, we'll talk about leukocytes and lymphocytes in terms of viral pathogenesis. That's what I'm talking about. You can always come back to this slide. Now, if you're interested in where these come from, many people study this. It's a really amazing series of events that start with a multipotential hematopoietic stem cell. Multipotential means can become many different things. They are self-renewing. They simply divide and make new ones of themselves. And as we'll see later, we've learned how to culture these from people, and we can introduce genes into them using viral vectors and put them back in people to restore things that are missing and so forth. Very exciting science. So these early on in development branch in two ways. They differentiate to become a common myeloid progenitor, which gives rise to all these cells on the left. Red blood cells, for example, are included here. But here are some of the leukocytes: basophils, neutrophils, eosinophils, monocytes, and macrophages. So that's the myeloid. They all differentiate from a myeloid progenitor. Then the other part, the early splitting of this pathway, gives rise to a common lymphoid progenitor, which, as you might guess, gives rise to NK cells and lymphocytes. So that's where these all come from, and many of them originate in the bone marrow. Now, last time we talked about how the dendritic cell senses when a virus infection is going on. The dendritic cells that patrol all over our body in different places, as we talked about later, are not active, but they do have receptors and mechanisms for sensing many things and then becoming active. They have toll-like receptors. They have MHC class II molecules so that they can display peptides. We'll talk about that today. They can produce cytokines. So these cells can detect viruses and viral proteins. They can detect cytokines. They can detect dead and dying cells. And they will react to these and differentiate and become activated. That's shown on the right here by the typical dendritic looking cell. When these cells become activated, a number of things happen in addition to the morphological change. The MHC class II molecules are displayed on the surface of the dendritic cell, and they contain peptides that are derived from the antigens that the dendritic cell has picked up. And this dendritic cell will be going into a lymph node to present those antigens to T and B cells, and if they're foreign, then the T and B cells will proceed along their developmental pathway. In the lymph node, these mature dendritic cells interact with T cells. That's shown here with B cells as well. So this is a CD4 precursor being shown interacting with the dendritic cell, but the DC can also interact with CD8 T cell precursors or B cells. And here we see the dendritic cell presenting a peptide in the context of MHC. So the MHC class II molecule is built to display short peptides in an antigen binding cleft. They're displayed in this case to the T cell receptor. If it's foreign, the T cell will then get activated. And basically, it's foreign if it's nothing self. As you go through early development, as your immune system develops, all the T cells that react with you are killed. So they're not present anymore. So you get rid of self, and anything that's not self is then considered to be foreign. It's an amazing thing that happens very early on in development. So this T cell is reacting to the presence of this peptide. If it's foreign, it will become activated and differentiate, in this case, into a helper T cell, which will then go on in adaptive immunity, as we'll see in a moment. The DC is also making cytokines which instruct the T cell on what to do. This is very important. The nature of the peptide and the cytokine often will be important for determining what kind of T cell are going to be made, especially if this is a CD4. There are a couple of different kinds of CD4 cells. The peptides and the cytokines determine which one it will be. Let's talk a little bit about antigen presentation. There are two main pathways that we have to consider. The one that we've just been talking about in the DC, picking up foreign peptides, is the exogenous antigen presentation pathway. And that, you can remember that because what we're doing is picking up peptides or proteins in the extracellular milieu. So here, this could be a DC cell, a dendritic cell, for example, picking up a protein or a piece of a protein that's been shed from a dying cell. It's taken into the cell by endocytosis. It is then fragmented, and then the peptides that are generated from that protein that was taken up are loaded onto the class II molecule in a vesicle, as those are the orange peptides there. Then the class II molecule is brought to the surface by the secretory pathway. This vesicle will fuse with the plasma membrane, and then the MHC class II is on the plasma membrane, on the cell surface, presenting a peptide. And on the right, that would be a T cell. So this could be a DC in a lymph node presenting a peptide that is derived from a protein that it's picked up to a T cell. And as I said, if it's self, if it's not self, the T cell will become activated. Now, as you might guess, there are many viral antagonists of this process, because this is not good for viruses, for the immune system to be activated and be directed against them. And one I've shown here is a herpes virus, human cytomegalovirus, which has been shown to interfere with the transcription of genes encoding MHC class II molecules. So in other words, this class II molecule is starting out in the endoplasmic reticulum. It's going to pass through the Golgi, but before it even gets there, it has to be translated from an mRNA. And this virus has specific antagonists that inhibit the transcription of that. What's the result? Well, the DC will not, or whatever cell is infected, will not be able to present peptides to an immune cell. So the existence of an infection will not be detected. And as you'll see today, there are many, many examples of interfering with all the pathways that we're going to be talking about. Here's one just, I'm telling you, for class II presentation. Now, so everything ends up in the lymphocyte, in the lymph nodes. So let's talk a little bit about lymph nodes. You have a very extensive lymph system there shown on this young lady who seems to be very unhappy that her lymph system is diagrammed, but it extends throughout your body. You have capillaries and vessels, and then you have lymph nodes, which are shown on the left. You can see the lymph nodes are the nodular parts on the diagram. On the left is a single lymph node, and it consists of a number of areas where both B cells and T cells develop and will encounter antigen. So material can be brought in by afferent vessels into the lymph node. It leaves by efferent vessels. And then, of course, there are arteries and veins to bring blood in and out. Now, typically in your circulation, about 1 in 10,000 to 1 in 100,000 B cells are specific for a particular antigen. It's not a lot. But if you have an infection and a DC brings peptides into a lymph node and they're deemed to be foreign, within a couple of weeks, you can have between 1,000 and a 50,000-fold amplification of those cells in lymph nodes. And that it causes the lymph node to swell. It's called lymphadenopathy. And if you've ever had a cold, a sore throat, you know that your lymph nodes right here underneath in your throat area swell. You can feel them. In fact, next time you have a sore throat, if you want to know if it's an infection or not, just feel your lymph nodes. And if they're not swollen, it's probably not an infection. It's probably something you ate or drank. But swelling of the lymph nodes is characteristic. Most of the time, you're not going to detect this. If they're deep lymph nodes within your body, you're not going to detect that they are swollen, of course. But if they're on the surfaces, under your neck, in the groin is another place where you could detect them as well. Next time you feel that, just think, those are B and T cells multiplying in response to a foreign antigen, and they're getting ready to make antibodies and to go out and start killing infected cells. Lymphadenopathy was one of the early signs of HIV infection. Before people understood what was going on, they noticed that the patients had swollen lymph nodes. The answer is E, all of the above. All of those are correct. Presentation of viral peptides on MHC2, endocytosis, activation by cytokines, and sensing by toll-like receptors. All right, let's take a look at the different effectors of the adaptive response. So we start here on the right-hand part. On the top are all the foreign proteins that can impinge on us, and we'll think about viruses. But of course, we could have anything, bacteria, parasites, fungi, any foreign protein coming into us would be recognized. We get infected by a virus. Virus is replicating in cells. Pieces of viral antigens are picked up by dendritic cells, and those are then presented in the lymph nodes either to T cells or B cells. So on the right is the pathway for T cell presentation. So here we have in the bone marrow, naive T cells which go through a developmental process in the thymus and then leave the thymus. They go into the lymph node where they're ready to take up antigen. And if the DC presents them something which is foreign, i.e. not self, they can either differentiate into cytokine-producing CD4 cells or cytotoxic T lymphocytes. So there's two different ways that this can go. There are two kinds of CD4 cells that can result, and which one is picked depends again on the peptide, the cytokines that are produced. They can be a T helper cell that produces cytokines that enable antibody to be produced by B cells. So we call those Th2 cytokines. Or the cell can become a Th1 cell, which makes cytokines that enable CD8 T cells to become functional. So you can see there are two main lineages of T cells, the CD4 and the CD8 cells, which are defined by a receptor on the cell surface. CD4, of course, happens to be the receptor for HIV, which we talked about a long time ago. So you can guess immediately that that's a target of infection. And the CD4s can differentiate into two kinds of helper cells, but basically their job is to make cytokines that either help CD8s differentiate or help B cells differentiate. The CD8 cells differentiate into cytotoxic T lymphocytes, CTLs. Their job is to kill infected cells, and they will do so by recognizing viral peptides on the surface of these infected cells, which are presented by MHC molecules. We'll talk about that in a moment. In the lymph node, antigens can also be presented to B cells, shown here. They become activated and eventually make antibodies. The activation is detailed on the left part of this slide here. Here is an immature B cell with antigen receptors or B cell receptors on its surface. You can see they look like antibody molecules, and in fact, if they engage an antigen, they stimulate a signal transduction pathway, which is shown by this jagged blue line, and that turns on transcription of genes that are involved in having this precursor differentiate into a plasma cell that makes tons and tons of antibodies. Grams of antibodies are secreted by these plasma cells once they respond to a foreign antigen. And the antibodies are these little red Y-shaped molecules, which are then the effectors that are going to have effects on virus infection. And we will talk about that. Also on the left here, you can see not only has this B cell engaged antigen and started to differentiate, but it's given help by a CD4 cell. This particular CD4 cell, which would be a Th2 type of CD4 cell, is making cytokines that help the B cell differentiate to a plasma cell. So basically, this helper cytokine arrow on the left here, that's the same as this green arrow going from the Th2 cell, helping the B cell become a plasma cell. So you can make, in response to a dendritic cell in the lymph node, you can make some kind of CD4 cell, which will produce different sorts of cytokines. You can make a CTL, and you can make a B cell, which will make antibodies. So let's talk about antibodies as in terms of what they do to viruses. I think most of you probably know what an antibody looks like. It's often schematized as a Y-shaped molecule. It's a tetramer, four polypeptides, 2 light chains and 2 heavy chains, and the circle is the immunoglobulin fold, which was first discovered in an antibody and now has been found in many other proteins as well. In 3 dimensions, these look very different here, but they do have a Y shape overall. The characteristics that are important, there's an antigen binding domain, so there's a hypervariable region of light and heavy chain, which will interact with antigen, and there are 2 combining sites per molecule, and that's called the FAB portion of the molecule. And then the rest of the molecule, which is more conserved, is called the Fc. And this has a variety of other functions, like it can bind to receptors on macrophages and help take up viruses and other particles into those cells. We'll talk about that more in terms of pathology later on. Now, when you inoculate an organism with a foreign antigen, you have an antibody response, and that's diagrammed on the graph on the right. So in this experiment, we're looking at serum antibody titer to some antigen that we've injected into an animal, and we're looking, for example, we've injected an animal with antigen A at the dotted line, and then within about 2 weeks, we start to have what's called a primary anti-A response. These are antibodies produced in the serum against that particular protein or antigen that we've injected. Now, this antibody response doesn't stay high forever. It eventually declines. The resting level is higher than the starting level, so you always have a low titer to whatever you're challenged with. So if I had measles years ago, I would still have a low level of measles antibodies circulating in me today. So you can see that diagrammed here. And the key here is shown if you then re-inject this animal with antigen A after 6 weeks or so, the second dotted line. You see you get a very rapid secondary anti-A response. You don't have a 2-week delay. It's rapid, and it's much higher than the primary response. That's the memory response to the antigen. And because so many things happen during the development of an immune response, it takes time. So for example, the production of high affinity specific antibodies takes a couple of weeks. And if memory doesn't require that time, because those cells that have been produced, those B cells that have been produced, which encode high affinity antibody against the antigen, will remain in you forever, and therefore you have a very robust secondary response. Now here is simply, at the second injection, they've also put in antigen B to demonstrate that there is a primary anti-B response at the same time, which again takes 2 weeks to generate, in contrast to the secondary anti-A response. There are a couple of different kinds of antibodies that we think about in terms of virus infection: IgG, IgM, and IgA. Now when you are, again, this is a experiment immunizing an animal, in this case with poliovirus, and then taking some serum at different times, days after immunization, and ask, are there antibodies to poliovirus present? You can see the first antibody to be made is IgM. It rises very quickly to high titers and then declines as the IgG titers rise up. Now, IgM is a polymeric antibody, shown in the upper left here. 5 molecules joined together by their Fcs to form a multivalent antibody. Why is this? Well, because the first antibodies that are produced haven't undergone all the somatic mutation and the rearrangement to get a really high affinity antibody. That takes weeks. So the idea is that the body initially cranks out the best antibody they can make. They make it IgM, and having 5 copies of it gives it basically higher avidity. It's low affinity, but the combination of 5 copies of the antibody makes for higher avidity. But you don't need that forever, so IgM levels decline. IgG molecules then rise, and these have been selected multiple times in the lymph nodes to produce or to be high affinity binders to the antigen. That again takes time. And IgG molecules are single molecules; they're not multivalent. Also, there's a third type of antibody which is very important, the IgA, which is secreted. You'll find these at your mucosal surfaces, respiratory tract, gut tract. They cross the epithelial layer and they're present in mucus so they can catch viruses before they even get into you. Very important for protecting us against respiratory and intestinal infections, for example. Now, the other consequence of this pattern is that if you're suspecting that someone is infected with a virus and you take some serum, you find IgM antibodies, you know you're pretty early on in the infectious cycle. If you don't find any IgM and you only see IgG against a particular virus, that means that the infection happened probably weeks or months ago. So IgM is a nice indicator of whether you're early on in an infection or whether you're quite advanced, because the IgM levels go down. You can see here after 2 months, they're gone. So if you have IgM, you're within those first 2 months at least of a poliovirus infection. Now, I mentioned that IgA antibodies get secreted, and this is a schematic of how that happens. So here we have a epithelial cell. It could be a respiratory epithelial cell. It could be a gut epithelial cell. Anywhere there's a mucosal surface, you have this epithelium, which we've talked about before. And in the basement area, the area below the basement membrane, the lamina propria, it's called, there are many cells floating around, including plasma cells. If plasma cells are produced, remember, they come from B cells that are activated in response to a foreign antigen, a viral antigen. They can go beneath this epithelial sheet. They'll produce antibodies, and among the antibodies they produce is IgA. IgA is produced as a dimer. It's called a polymeric IgA, pIgA, which is linked covalently. That IgA, in order to get to this surface, remember the brush border here is the lumen of the respiratory tract or the gut tract. That antibody has to bind a protein that gets, it's called the secretory piece. It's shown here in brown. It binds the IgA. It's then endocytosed. It's transported by transcytosis to the top of the cell. It's then released, and the antibody is cleaved from the surface of the cell, and it ends up being in the mucus that's lining the top of this cell. So that's how IgA gets from the bottom to the top. IgA, as you saw from the graph, takes a long time to come up, and it goes away very quickly. And this is, in many infections, respiratory and gut infections, IgA is very protective, but it doesn't last. And so that's why we often get reinfections with a number of viruses that target those sites. Now, what do antibodies do? Some of them are what we call neutralizing antibodies. They're a subset of all the antibodies that are made against a virus, which have the ability to block infection. That's what neutralization means. It stops infection. All right? And we'll talk in a moment, some mechanism of how antibodies block infection. But you can imagine, and you make an antibody response against the virus, that those antibodies can bind up virus particles and block their infection, and that can stop infection. Now, it turns out that antibodies are essential defenses against many virus infections. That's why we immunize against many virus infections. That immunization generates antibodies and memory, which will then protect you if you are ever infected with the real virus. In some viruses, the antibodies neutralize particles in the blood, so it can prevent spread from a primary to a secondary site of infection. As I've just told you, a secretory IgA at mucosal surfaces blocks entry before the infection even gets started. So one of the polio vaccines that we use actually blocks infection in the gut and prevents replication. So most antibodies, as far as we know, are essential defense. They prevent infections, but they're not great at resolving infections. Because after 2 weeks, it takes 2 weeks to get a nice titer of IgG. By then, many viral infections are over. And that's because recovery often depends upon cellular responses to virus infection. However, like everything else in biology, it's not absolute. Some antibodies turn out to be important for recovery as well. So here's the demonstration of an assay to show neutralizing antibodies. It's the plaque assay, which we encountered a long time ago. And you remember, plaque assay uses monolayers of cells, and wherever a virus is infecting, you see a circle, which is a zone of killing of the cells. And what we have here are two different samples of serum from animals. One animal has been immunized with a virus, and the other has not. So the one on the bottom has not been immunized. It's a control animal. And what we do is we mix a little bit of virus, enough to give a dozen plaques or so, with dilutions of serum from these animals, and then we do a plaque assay. So you can see at a low dilution of the serum, so the number here is a dilution of serum, 1 to 10. There are no plaques. 1 to 100. 1 to 1,000, now you're starting to get some plaques. And 1 to 10,000, there's no more neutralizing activity. So you can see the neutralization effect is dilutable, which you would expect if it's a soluble substance. And of course, these are antibodies that we are diluting out. And we simply have enough of them so that at 1 to 10, 1 to 100, they block all the infectivity. The serum from the control animal has no neutralizing activity at all. We could do this experiment with your serum. We could take a little bit of your serum, and then we could take a number of viruses and see if you have antibodies against them. And that you would use an assay very much like this. So that's what a neutralizing antibody is. It blocks infection of cells, either in tissue culture dishes like this or in an animal, and that's how they work. Here's an experiment that shows you the protective effect of an antibody. This is a passive experiment where we're giving animals serum from another animal that's already been immunized with, in this case, poliovirus. And then we take these animals, we have different groups of animals, we have a control, and then we have 4 other groups that get different amounts of antibody. So we have antibody in the form of serum from an animal that's been immunized, and we're giving it to a second set of animals, and we're diluting it out. So the control animals do not get passively administered antibodies. We then challenge these animals with poliovirus, and we're looking at the percent animals paralyzed. So the control animals, 100% of them are paralyzed because they have no protection against poliovirus. They didn't receive passively. Passively simply means we inject into them. They didn't passively receive antibodies from the immunized animal. If we have the titer of 1/3, which is a relatively low titer, we have no protection either. But as we increase the amount of serum essentially from the immunized animal, you begin to get protection against paralysis until at this very high titer, all the animals are protected. So it's an example of how antibodies can block infection if they are there already. So if you had to wait 2 weeks in order to induce these antibodies, they wouldn't protect you. You could recover from infection, but they wouldn't protect you. But if you immunize an animal, you can protect them against infection. Or if you give people or animals antibodies, preformed antibodies, you can protect against infection. Now, we usually don't give people preformed antibodies because they only last for a few months at best, and you'd have to go back and do it again. Only in emergencies do we give people preformed antibodies. For example, if any of you were bitten by a rabid animal, you would get preformed antibodies to rabies virus while you were being immunized against rabies virus. Very few of you are likely to be immunized unless you're a vet or have worked with animals and so forth at risk. So if you do happen to encounter the virus, we give you antibodies to prevent spread of the virus while you're being vaccinated. During the Ebola virus outbreak a couple of years ago in West Africa, there was no way to stop infection. Eventually 25,000 people got infected, but we did have some antibodies against Ebola virus. You may remember the famous ZMapp is the name of one of them. It's actually a mixture of 3 different antibodies. They gave that to some people to try and cure their infections. So that's the basis for that approach. So how do antibodies work? So neutralizing antibodies in specific. Not all antibodies produced against the virus can actually neutralize infection. Only a subset of them. They're not against the right antigen. They will perhaps bind to the virus but not block infection. But the ones that do block are called neutralizing. Here are some mechanisms. We're using rhinovirus as an example. So on the left, we're showing the early replication cycle of rhinoviruses binding receptors. It's being taken into the cell by endocytosis. The pH of the endosome is dropping and the RNA is getting out. So antibodies on the right are shown interacting with rhinovirus and blocking infection at different steps. So obviously, if you have enough antibodies bound to the virus particle, you could block attachment. So that could certainly be one mechanism of neutralization. It's not the only mechanism, though. There are others. Some antibodies seem to be able to block endocytosis, the uptake of the particle. That's number 2. Some can block uncoating. They can somehow lock the virus so that even at the low pH, it never opens up and releases the nucleic acid. Some viruses somehow block attachment. Number 4, neutralization after replication starts. Some antibodies have been shown to get into the endosome with the virus particle, and then after the virus is released in the cytoplasm, these antibodies seem to be neutralizing infectivity. And so finally, number 5, some antibodies can cross-link virus particles. Remember, there are 2 binding sites for the epitope on each antibody molecule, And so you can imagine that they could link two virus particles together, and you could get quite a big aggregate, which simply isn't effective at getting into cells. So those are different ways that neutralizing antibodies can prevent infection. But those are not all the ways. There are even more, which are shown on this slide. So again, we have a virus particle. We have neutralizing antibody bound to some epitope on the particle. I've just talked to you about neutralization of infectivity, 5 different ways that antibodies can block infection. But antibodies could also lyse enveloped viruses. So we really briefly talked about the complement system last time, but the complement system will recognize antibodies bound to an antigen in a membrane and induce the lysis of that membrane. Complement-mediated lysis. So you can imagine if an antibody is bound to an enveloped virus, complement, which is a collection of proteins in your blood already there, doesn't have to be induced, could lead to the lysis of the, lysis of the particle. And of course, that particle would no longer be infectious. But even after infection has begun, sometimes antibodies can work. So here, for example, is an infected cell on the bottom. And this cell is pretty far along in infection such that viral glycoproteins are being produced on the cell surface. Well, antibodies could bind those and lead to complement-mediated lysis of the infected cell. And there are a number of other mechanisms shown at the right here. So we've just talked about lysis by complement. Sometimes a cell with an Fc receptor, so this part of the antibody sticking out into space here is the Fc portion. Some cells have Fc receptors, so they'll recognize this cell as bearing an antibody and lyse it. So that's Fc-mediated effectors. The antibody binding can lead to signaling pathways that inhibit virus replication or release or even cell-to-cell transmission. So there are lots of mechanisms of antibodies interfering with a virus infection, not just binding to virus particles at the early stages of infection. So here is a lovely picture of poliovirus bound to a particular antibody in purple. So the antibody is in purple. Now let's see if you remember anything from the early part of this course. Where in the virus particle, this is an icosahedral particle, where in the virus particle is this antibody binding? The 5-fold axis, right. Look, 1, 2, 3, 4, 5 antibody molecules are bound at each 5-fold axis. So that's where the epitope is for this particular antibody. It's at the 5-fold axis. Right there in blue in the center there would be one 5-fold axis. And of course, there are a dozen 5-fold axes in the particle, and so each one is decorated with antibodies. So you can see if the particle were saturated with antibodies, and this is the virus, the receptor binding site, it would be hard for these particles to attach to receptors. Now, do you think that viruses can evade antibody responses? Remember, I told you, always answer yes when I ask you an immune evasion question, always. Absolutely. And here are 2 general strategies for doing that. So on the left, rhinovirus. Rhinovirus, there are over 150 different rhinoviruses that circulate globally. And if you're infected with one, you're not going to get infected with that one again, but you could get infected with the other 149 rhinoviruses. So I view that as an evasion strategy, the generation of sufficient antigenic diversity such that there's so many different types circulating that you can't be protected. That's why you get colds almost every year, because there's so many circulating rhinoviruses. Not every virus can do that. It's very rare. Measles virus, there's 1 antigenic type. Poliovirus, there are just 3. Why those viruses can't vary like rhino can, we don't really understand. But very few viruses can do that. As we'll see later, HIV is very good at evading antibody responses after it infects you. HIV infects you, you make a wave of neutralizing antibodies, and the virus changes. By the time those 2 weeks are up and the antibodies are made, virus has changed. Okay, so let's make another set of antibodies. Virus changes again. And this goes on. And of course, at the same time, the virus is infecting CD4 cells, so your ability to make antibodies gets lesser and lesser. Anyway, so we'll talk about that in more detail later. So that's rhinovirus's strategy. On the right is a strategy of influenza virus. This is the hemagglutinin, which is one of the glycoproteins in the virus particle. Remember, it's a trimer, it's a stalk-like molecule, and it's this molecule, of course, attaches to the receptor on the host cell. Down at the bottom here, the HA would be attached to the virus. The sialic acid binding site, which is the receptor on the cell, is at the top. And all these regions in color are different epitopes of the HA. In other words, you make an antibody against the HA, and each antibody is only specific for a very short amino acid stretch called an epitope. And then you can map all the epitopes on this protein that are bound by antibodies. And they're shown here in color. The ones on the top here, single amino acid changes define some of these epitopes. As this virus is replicating in us, those sites change and it evades the binding of antibody. These sites on the stalk, the purple and the red ones, are highly conserved and they change much less. So in the course of a single flu season, these top epitopes, the orange, green, blue, yellow, magenta, those can change, and one amino acid is enough to prevent the antibody from binding to that epitope. And that's why often we change the flu vaccines from year to year, because the virus has sufficiently changed. It only needs one amino acid change in these epitopes to evade the antibody response. So that's another mechanism of antibody evasion, antigenic variation. Some viruses do that, again, but not all viruses. Next question is, which statement about antiviral antibodies is incorrect? They're important for protection against virus infections. They only neutralize virus infectivity. They may block virus attachment. They can be found at mucosal surfaces. IgM is the first to appear, then IgG. The answer is B. Very good. What's wrong is that they only neutralize virus infectivity. They do things after the virus has already infected the cell. And why A is wrong? Well, the basis of vaccination is antibody, right? You immunize people, they're protected against infection. So that's clearly not wrong. That's antibodies. Let's talk about cell-mediated immunity. Now, in contrast to antibodies, and again, this is not absolute, there are exceptions, but in general we say the cell-mediated immune system is essential for clearing most virus infections, opposed to antibody. And what happens is, this comprises, of course, CD8 T cells, which we call cytotoxic T lymphocytes, or CTLs. These form what we call an immunological synapse, which simply means that that CTL comes very close to an infected cell with a very tight junction in between them, and it lyses the target cell. All right, and of course, we'll explore this in mechanistic terms in a moment, but the idea is that the CTL is recognizing a cell as foreign or as bearing a foreign peptide in the MHC1 molecule. Of course, if this peptide is not foreign, the CTL will not kill the cells. And of course, there are countermeasures against cell-mediated immunity, as we'll see in a moment. Now here, MHC1 is a key player. MHC1 is how CTLs will recognize the peptide presented by any number of our cells. So MHC1 tends to be on all of our different cell types, as opposed to MHC2, which is on specialized cell types. So here is MHC1 pathway, which we call the endogenous antigen presentation pathway, as opposed to the exogenous pathway, where cells, specialized cells like dendritic cells, are taking up foreign antigens. Here, the antigens come from within because the cells are infected. These are the target cells of the CTLs. So here we have a virus-infected cell. And of course, the infection is making proteins, viral proteins, and these are put through the garbage disposal of the cell, the proteasome, and chopped up into small peptides, which are then loaded in the endoplasmic reticulum onto the MHC1 molecule. And then those molecules are transported up to the plasma membrane by the secretory pathway, and at the cell surface, the MHC1 is presenting the peptide, which is shown in orange. There's a higher magnification view at the right there. And then we have a cytotoxic T lymphocyte, which is coming up and asking, is this a foreign peptide? And if so, the cell will be killed. Now, of course, that causes damage. You're killing a cell, but the idea is you stop virus replication and maybe you spare the organism. Now, there are lots of examples of viruses antagonizing this pathway. You can imagine that there would be, because if you're a virus-infected cell and you're eliminated by a CTL, that's the end of the infection. So herpes viruses are very good at this. The cytomegalovirus, we saw already, antagonizes MHC2. Herpes simplex, Epstein-Barr virus. And as we'll see in a couple of lectures, these are viruses that tend to cause long-term persistent infections. And they're good at doing that in part because they can evade killing of infected cells by CTLs. So all the circles here are targets for antagonism of the MHC1 pathway. So for example, some viruses make proteins that inhibit the proteasome, so it can't degrade their proteins and make the peptides. The peptides are brought into the ER lumen by a protein called TAP, the transporter associated with antigen processing. You just think of it as a transporter that brings the peptides into the ER. There are inhibitors of TAP, viral inhibitors. There are inhibitors of peptide loading onto the MHC. There are also inhibitors of assembly of the proper MHC molecule, as you'll see. And so there are lots of ways. And all of these mechanisms would end up having no peptide in MHC1 on the cell surface, so the cell would not be recognized as foreign or infected, and it wouldn't be killed. So these are good ways to avoid elimination of infection. So here is just a table showing you these mechanisms in some detail. Countering MHC1, so that's the picture I just showed you with the red circles. So we have different parts of the MHC1 pathway. For example, the synthesis of MHC1 can be inhibited by a lentivirus protein, an HIV protein called VPU. The synthesis of the transporter, the peptide transporter, or its function are inhibited by a variety of herpes virus proteins. You don't need to, of course, know any of these names. You simply need to know that you can interfere with MHC1 at multiple steps. Transport of MHC1 to the ER, that can be interfered with. Some viral proteins cause the MHC1 to be retained in the ER, so it can't go up to the cell surface. They keep it back there by binding to it, and those proteins have an ER retention signal. So they stay in the ER, so obviously that's an evasion. Sometimes some viral proteins cause the MHC1 to come off the ER into the cytoplasm, so they never make it up to the plasma membrane. And if the MHC1 should make it to the plasma membrane, there are some viral proteins that make it get taken back up into the cell. They increase the endocytosis. So almost every step of this MHC1 pathway, you can get antagonism. How does the lysis occur? So CTLs recognize a virus-infected cell by this foreign peptide presented to them in the MHC1 molecule. That's shown here on the left again. There are two mechanisms. In both cases, the CTL insert into the target cell a pore. And you can see there are two kinds of pore here. There's a lytic pore on the left. And what this does is basically allow all the contents of the cell to come out, and it kills the cell by releasing the cytoplasm. So that is the release of cytoplasmic content mechanism. The other mechanism of killing is that the CTL makes a pore in the target cell membrane. Again, it's a different kind of pore. It's called a perforin pore. And then it injects a protein called granzyme B into the target cell. Granzyme B converts procaspases to active caspases, which leads to the induction of apoptosis, programmed cell death. So that basically the granzyme leads to the death of the infected cell. So two different mechanisms, and that's how CTLs kill a cell that it detects to be infected by virtue of that peptide presented to it in MHC1. Remember, if there's no MHC1 on the cell surface, there's not going to be any killing, and that could be a virus-infected cell that's somehow evading the presentation. This is a series of experiments which show the kinetics of CTL production in an infected animal. This is an animal that's been infected with a particular virus, and at different times infection. In the top panel, we're simply looking at virus titer in the serum by plaque assay, or this is PFU per spleen, excuse me. So we're taking out the spleen, we're grinding it up and asking how many PFU are in each spleen. And so you can see there's a peak of viral replication in the spleen before 10 days post-infection, and then the virus in the spleen rapidly declines. And that declining is probably due to lysis of infected cells by the CTLs. On the bottom, we're looking at CTLs, cytotoxic T lymphocytes. We're using two different assays, and they're represented on the two y-axes. So on the left y-axis, we're looking at effector CTLs. These are simply assays where we're asking if we take T cells, CD8 T cells out of the infected animals, will they lyse in a tissue culture dish target infected cells? Okay, so we're assaying for their CTL activity. So that's CTL, and that's in green. So you can see very rapidly after infection, you have a very large number of cells that are able to kill virus-infected cells, CD8 cells. The other way these CD8s are assayed is by their specificity for epitopes. You can ask, are we generating, or is this infected animal generating CD8 T cells that are specific for viral peptides? We don't ask anything about lysis here. Simply, do they recognize viral peptides? And you can see that also goes up very quickly. It peaks coincidentally with the effector CD8s, but those stay around for a long time after infection. So the epitope-specific CTLs can last a long time. And again, these are very important for recovery for infection in many virus infections. So let's do an experiment where we look at the role of antibody versus cellular immunity in a particular model. We're looking at monkeypox infection of monkeys. And we do 3 different manipulations in this experiment. So we're going to immunize these animals with monkeypox inactivated monkeypox virus so it doesn't kill them. And then the control animals are on the top line. By day 22, we can demonstrate high levels of neutralizing antibodies in the serum of these animals. So we take some serum, we do a neutralizing plaque assay test, as I've shown you, and these are very high titers. These are basically the reciprocal of the dilution you would need to get rid of neutralizing activity. We then infect these animals on day 28 with monkeypox, and all four of them are protected against infection. So antibody seems to be correlating with protection. So to prove that, what we do in the second group of animals is we deplete them for B cells, which we can do with an antibody. If these were mice, we could do it genetically. By day 22, you see very low neutralizing antibodies. Not surprising, as we've taken out all the B cells, which are the source of neutralizing antibodies. We then challenge the animals on day 28, and 3 or 4 of them are dead. So only one has managed to survive without antibodies. And finally, we can take a third group of animals, deplete their CD8 cells. We get good levels of neutralizing antibodies by day 22. Challenge them. None of them are dead. So this clearly shows that cellular responses are not important in protecting these animals against infection. However, if we continued longer and asked how they recover, we might find that cellular responses were important for that. So this balance between CTLs and antibody tends to vary from virus to virus. For some, CTL response is more important. For others, antibody is more important, even for protection. How is it determined? It really starts in the lymph node when the DC is in there giving peptides and cytokines to the B and T cells. It's the combination of those that determines whether we're going to have a CTL or an antibody response predominating. And that decision is helped along by these subsets of CD4 cells, the 2 different TH cells or T helper cells, TH1 and TH2. So as I've said, in the lymph nodes, these TH cells are making contact with the DCs and the macrophages that have brought in peptides and are presenting them to them. The DCs, remember, are presenting the antigen to these cells. They're also secreting cytokines. It's the mix of the peptide and the cytokines that determines what kind of outcome you're going to get. So here on the right, we have an antigen-presenting cell. It has detected a viral antigen by a toll-like receptor. It's making IL-12, and that leads to the production of TH1 helper cells. And this will favor the production, which in turn favor the production of CD8 cytotoxic T lymphocytes. So again, it's the particular combination of the peptide detected in the cytokines that are produced that give rise to CD4 cells that are going to help in the development of CTLs. In this case, we have not favored the production of antibodies. We have not favored the production of Th2 CD4 cells, which would make antibodies that would neutralize virus infectivity. This whole matrix is diagrammed on the left. So at the top, we have an antigen-presenting cell plus an immature CD4 helper cell. And this is going to happen in the lymph node. And then depending on the peptides that are presented and the cytokines that are released, we can either differentiate to Th1 cells, which, again, favor the production of CD8 T cells. And the cytokines that favor their production include IL-2, 12, and interferon gamma. On the other hand, certain viral peptides will stimulate the production of IL-4, 5, and 10, which give you Th2 CD4 cells. And they, of course, produce cytokines that allow B cells to differentiate. So the particular peptide plus the combination of cytokines that are produced in the lymph node by the dendritic cell or the macrophage presenting the antigen will determine whether we're going to ultimately have a CTL or an antibody response or some mixture of the two. I should also say this, there's some cross-regulation here. So as you make, for example, cytokines that that stimulate the production of, say, Th1 cells, these will also inhibit the production of antibody stimulating Th2 cells. So that's where this all happens very early on in the lymph node. Our last question, for some infections, CTLs are more important for protection than antibody. How is the CTL antibody balance determined? By toll-like receptors, by intrinsic defenses, by autophagy of infected cells, by the mix of peptides and cytokines presented by DCs. It depends on whether the capsid is icosahedral or helical. Answer is D. The mix of peptides and cytokines presented by DCs. It doesn't depend on the nature of the capsid, of course. Now, these adaptive responses also are associated with memory. As I showed you before, it takes a long time to make antibodies when you're first encountering a foreign antigen, but the second time, the response is rapid and specific. And this distinguishes, of course, adaptive from innate. Innate responses don't have memory. So immune memory is important, and it's all a matter of timing and extent. The response is a lot quicker in a secondary response, and so the antibodies are present to prevent infection, whereas if you need 2 weeks to make antibodies or more, you have no way of stopping most infections. And of course, the magnitude of the response, the antibody response, is typically much higher after a memory response as well. And that's why we vaccinate people, because once you're immunized, you have a memory of the immunization, and if you're challenged 20 years later with whatever virus you were immunized against, you will be protected. So I got a measles vaccine as a kid, and I was less than 10 years old. And if I were challenged today, I would not get measles. I would still be protected. And it wouldn't simply be those low levels of antibodies in my serum, it would be the memory response. So there's a nice experiment done a long time ago by an epidemiologist, which before we even understood anything about viruses or immunology, showed that there is immune memory. And this is a study done on the Faroe Islands, shown here on the left, to the north of Scotland. And in the 1700s, they were isolated. The only way things got to the island or out was when ships came, airports back then, right? And so people were isolated for many, many years. In 1781, there was an outbreak of measles on the island. Probably it was brought in by someone arriving in a ship. But there was an outbreak, there was recovery. Next 65 years, no measles on the island. I don't think no ships came for the next 65 years, but they were lucky that no one came with measles on any of those ships. 1846, there was another outbreak of measles. None of those who had survived the 1781 epidemic and were still around were infected. There were other people infected, but none of those individuals. And this epidemiologist noted this. He said, there's some kind of memory going on here, and now we know, of course, that this is immune memory. It lasts a long time, and you don't have to have re-exposure to the virus. For many years, people were thinking, maybe memory needs constant low-level exposure to the virus. But it's not true, because there were no other outbreaks of measles in the intervening years, so we know that's not the case. So here's the summary of memory in a graph format. We're looking at time on the x-axis, and on the y-axis, antibody or T cells. So memory happens in both antibody producing and T cells. We have our first infection, and again, by 2 weeks, we have an initial antibody response, too late to control the infection. But then our immunity wanes. For all these years, we have protective immunity, a low level of antibodies in our serum. And then, say, years later, we are challenged with the virus again. We have a very rapid production of antibodies. We have higher levels of antibodies produced. And now it happens so early in infection that it's able to prevent infection. Again, at the same time, the T cells are being elaborated. They also take time to produce. And they're ready by the time the infection is full-blown, and they can start interfering with virus production and can actually help in recovery. So that's the basis of immunological memory. So memory basically consists of cells which stick around for a very long time. They're long-lived cells that probably reproduce very infrequently, and they include what we call memory B cells. So remember, when a B cell precursor becomes activated, it becomes a plasma cell. These make tons of antigen. Eventually, the plasma cells go away. You don't need them to produce so much antibody, but you're left with some B cells that have differentiated to produce the specific antibody, high affinity antibody specific for your antigen. That requires a lot of somatic mutation and rearrangement. Those remain, and those are called memory B cells. They hang around in the spleen and the lymph nodes. They don't make antibodies unless they're stimulated by antigen, unless you encounter the virus again. There also seem to be some long-lived plasma cells that just stick around. Not too many of them, but a few, and those are ready to make antibody as well. They tend to be in the bone marrow. And there are also memory T cells. Again, memory T cells that have differentiated to be specific for that peptide that is produced and presented in the infected cell. They will stick around. They're long-lived, and they can respond very quickly when a new infection occurs. So let's try and make an overview of all this. As we're done thinking about the immune response in a kind of didactic detail. We're going to be using it, of course, in subsequent discussions to understand how disease is prevented and how it occurs. It has a role in both. But let's now put it together in different body places and see if we can understand what's happening. So, different parts of our body have different sorts of immune systems in terms of specific names of cell types and so forth, but the concepts are the same. Here is your skin. We have a cutaneous immune system in our skin, and remember, the outer layer of our skin is dead, as shown by the lines here, but below it, the epidermis is full of cells, and here at the bottom is a blood vessel. Within the epidermis, there are a number of different cell types that can participate in immune responses if a virus is introduced into this area by a scratch or a needle or a mosquito and so forth. So we have keratinocytes, which are a major type of cell in the epidermis. They produce keratin, the characteristic protein of the skin. They have a lot of immune functions available to them. They have class 1 and class 2 molecules, so they can take up and present molecules. Sometimes they migrate out of the epidermis and go into the blood or go into a lymph capillary and present these antigens to lymph nodes. They can also make cytokines and initiate an inflammatory response. There are also a subset of dendritic cells called mobile Langerhans dendritic cells. And these are very much like the dendritic cells we've talked about. They're present in an inactive form, and if they encounter cytokines and antigens from an infected cell, they will become differentiated. As you see here, they will produce cytokines, and they can go to the lymph node also and instruct the T and the B cells. And presumably there's a neighboring lymph node not too far away, and that if the T and B cells are activated there, they would go back to the infected area. We also have T cells of various sorts, mainly CD4 T cells in the epidermis. You can see both Th1 and Th2 cells. So these can begin to respond to viral peptides immediately right in the skin without waiting for the lymph nodes. So that's an example of a cutaneous immune system. This is something that would protect your skin from infection. The mucosal surfaces are also vulnerable, as we talked about very early on, the respiratory tract and the gut tract. So they have their own set of problems. This is the particular mucosal immune system in the gut. So here we have at the very top the lumen of the intestine, and we have our layer of epithelial cells with our brush border. And of course, on top of that would be a layer of mucus. If there were respiratory tract, there would be a lot of mucus there, but there's some here as well. And then we have in the lamina propria, which again is just the space immediately below the basement membrane, you have a large, extensive immune system. So here you can see blood vessels as well as lymphatic vessels. Of course, cells can go into the lymphatic vessel to reach the neighboring lymph node. There are large collections of lymphocytes throughout the gut. It's called the gut-associated lymphoid tissue, or GALT. Here's one of them in blue. It's called a Peyer's patch. Lots of different lymphocytes in there. And these cells, called M cells, are constantly sampling the antigens in the lumen of the gut and passing those antigens onto T and B cells below. So there's sort of a localized lymph node, if you will, which is much smaller than a real lymph node right in the gut. But we have all sorts of T cells here that are ready to respond to antigen as well. Here's an antigen-presenting cell, for example, presenting an antigen to one of these T cells, so they could become activated and begin to secrete cytokines here, or they can move to the local lymph node. We also can have plasma cells that have either differentiated here or come from a local lymph node, producing antibodies, which would be available in the subepithelial space, or that could be secreted as secretory IgA as well. So every area has little differences such as these, but you're basically patrolled very extensively by your immune system just to be ready for virus incursions. And I want to end up by emphasizing how important inflammation is in coordinating the innate and adaptive responses. So last time we talked about innate responses, today adaptive, and they're linked by inflammation. Inflammation is a consequence of a good innate response, and it sets up the adaptive response to be strong. And remember last time we said viruses that don't cause inflammation are not good at making antibody responses. And that's because the inflammation helps the innate response to communicate with the adaptive response. Parts of what's happening in inflammation, cytokines are being produced that recruit cells to the infected area to synergize and amplify the adaptive response. So here we have a hypothetical infected area. So where you see antigen there would be a virus infection in this area. It could be a subepithelial area, it could be almost any tissue. But what happens when viral antigens are being produced? They can be sensed, of course, by dendritic cells. They can be picked up early on by dendritic cells, who would then go to the lymph node and instruct T and B cells. Eventually, those cells would come back to the infected area. They would be brought there by cytokine gradients that are produced from the infected cells, so they know where to go. T cells killing cells, B cells making antibodies, as we discussed. But even before that happens, the antigen can activate complement. Complement proteins are always present in tissues, and complement activation in a variety of ways can lead to increased phagocytosis of viruses, for example, and the production of all the complement proteins that are chemotactic. They can attract cells from the blood into the infected area. They can lead to the production of chemokines in certain cell types as well. And a lot of these compounds that are made, the cytokines and so forth, the chemokines, they could, for example, make the bloodstream more permeable to allow cells to come out of the blood and go into the infected area. They could even bind to receptors and allow lymphocytes inside the blood to attach to the endothelial cells to roll and to go through as well. We talked about that last time. So all of this is an interconnected system. It begins with sampling of foreign proteins, the production of cytokines, the activation of complement, and again, in the end, if the infection is not controlled, the T and B cells will come in and produce their effector proteins. So keep this all in mind now. The immune system is here to protect us against infection. It doesn't always work, as we'll see, very well, because viruses can overwhelm it. But also remember, a lot of the mechanisms we've talked about cause damage to us, starting with the interferons, which cause their own kind of damage. Antibodies can be pathogenic, and obviously cytotoxic T cells as well. So in the next few lectures, we're going to be talking about the mechanisms by which viruses cause disease, and that'll play a really important role in that.